speaker to be Jocelyn Etienne from Grenoble. Can you tell us about cells and embryos?
Thank you. So, well, first I'd like to have a word to thank the organizers of this workshop and this whole program, because I think it's been a great opportunity for all of us to meet many people. And as you can see from my first slide, I quite like meeting many people and discussing things, and I think that's really one of the most important steps of research, is actually meeting the right people and start working with them and start talking about their systems and so on. So you see many systems here. You see single cells in this biophysics group in Paris, led by Atef Esnesios. I presented something on that in September. You see here, these are actually cells which are tracked in an embryo in early Drosophila development in the group of Bénédicte Sanson here in Cambridge. So these people in Math Institute in Grenoble are doing, developing numerical methods along with me for these problems. And then these colleagues from my institute in Grenoble have been working on shells and buckling and shell deformation. So I'll try to put all that together to give you an idea of why I think we can consider cells and embryos as flowing shells in the case for as long as the actomyosin-based mechanics is concerned. Then we will try to write a mathematical model for that. That I will be able to study in some limits. So one limit is the elastic limit. This will be a viscoelastic model, so of a liquid. A viscoelastic liquid is elastic in the short times. So this is the elastic limit for the short times. I'll also be able to solve the viscoelastic equations in a simple geometry and show you also some calculations of in the viscous limit this time, so long times. So why do I claim that viscoelastic liquid shells are appropriate for the problem? So these are recent images obtained in super resolution microscopy for cells, and now we're beginning to see really the 3D shape and structure of the cells, which was something which was missing just a few years ago. So here you see really that this is visualization of the actin. So you see it from the dorsal side. That's the type of image we've had for many years now. But here you see really that it's located in a very thin layer at the periphery, although there are also structures here which are crossing the cell. And then another image of another cell type, and this cell is also, this snapshot is from the cell when it's still spreading and not polarized yet. So here it's polarized, there is a preferential direction. Whereas here it's not polarized, and you see it is pretty symmetrical, and again you have a structure which is really at the periphery of the cell. So this leads to this type of modeling geometry where you have a network of actomyosin at the periphery interacting with the substrate through adhesions. And in the middle you have something else, but mostly not actomyosin. And we know that what gives this shape will be the thin structure here. Now argue that you have to treat this as something flowing, because if you look in the same paper, when they look at the actin and myosin, so this is the myosin actually, which is not, can we have the light a little less bright in the front, please? Hello? Could the light be turned off?
Yeah, they will.
Okay. Yeah. So I'll play that again. So you see that this retrograde flow of the myosin here, which is actually bound to actin. So you have these things flowing together. So you really have a flowing shell. Okay, one more time now. Yeah. So now you probably see it better. And we've seen similar images in the talk by Sasha Bershetsky this summer. Okay, and then when we have to, if we want to understand the shape and the mechanical equilibrium of this, we have to worry about the stresses. So if we have a thin structure, we have a stress tensor, a tangential stress tensor with two directions and possibly some shear there, which is living on this thin surface. And we see, for instance, we can measure the forces that this structure is exerting on the substrate. This is traction force microscopy with actually the forces in the plane, and there is a color code also for the forces going up at the periphery and the downward forces in the middle, as you would expect for something which is probably pressurized also by this tension at the periphery. And now this tension and this shape is actually controlled by the activity of myosin. So here are three different ways of controlling the myosin activity in this cell, still in the same paper. So in the control cells, you see where the myosin is active, you see that there's a very flat lamella at the periphery. And then if you downregulate myosin in three different ways here, you see that you go back to a shape which is much closer to the spherical cap that a droplet with a constant surface tension would assume. Okay, so you really have a control. I'm not saying how this is happening, but there is a control by the myosin of this shape. And actually these cells which don't express myosin normally, if you express myosin in them, so this is a different cell type here, if you add myosin into them, then you obtain this flat lamella that you had in the control cells of those expressing myosin endogenously. Okay, now I said cells and embryos. So this is an early Drosophila embryo. So you have this ellipsoidal shape. This is the same one, dorsal side and ventral side. And you'll see what happens to this guy. So you have first the ventral furrow formation. That was the lips that you were seeing. And then the germ band extension going this way and around the posterior end of the embryo flowing upwards here, and there is something else which is called midgut invagination. So many morphogenetic movements happening in that. What you see here is the nuclei of the cells, which are located close to the external surface, to the apical surface of the cells, and in the middle is the yolk, is the viscous liquid. So is this a shell? Well, it must be believed that what is really driving the flow is the movement of the apical surface, the external surface of the cells, which are here tracked and color-coded so that you can follow them. And you see really this surface flow of these cell apices. And this is done by the collaborators in Cambridge here. Okay, and it is mostly assumed, and I'll argue also why this should be considered as a flowing shell. So the question I'd like to address is, if I'm given the structure of these objects and the location of the myosin on them, can I predict really why they are at mechanical equilibrium in the shape they have? And what is this mechanical equilibrium? Can I characterize it? And can I predict the short-term deformations of them? Of course, then, so that's really going from this patterning of, say, we have some gene being expressed in this region, which turns into expressing myosin there. Can I get this flow? And then, of course, there would be further questions. Once I have this flow, this will have an influence again on the gene expression and on the location of the structure of the structural elements. But let's focus first on this, and for the coming years we can focus on
that. Is this in 2D or in 3D?
So this is 3D. This is a surface of the embryo.
And the forces you present are in 3D.
And here this is the flow. This is the tracking of the flow in an experiment. And they have tracked it actually in 2D because they didn't have visualization in 3D.
That's what I'm puzzled about, because...
But now we are beginning to... So that's very recent, this type of tracking in 3D. And so from these data, we can have the velocities in 3D on the surface of the embryo. So that's beginning to come, but that's also related with super resolution microscopy. That's quite new. Okay, so let's start and write the equations for this. So first, we have no inertia. Okay, it's the Reynolds number cannot be larger than 10 to the minus 5, even in the embryo and in the less viscous flows there. Then we need to balance the surface forces. So we have a surface divergence of this stress tensor, which is living on the surface, plus the bending forces, the bending rigidity forces of this thin shell, which must balance at least the pressure difference between the exterior and the interior, which is possibly submitted to some drag forces of the fluids inside and possibly outside of the object. And there are also forces that can be exerted directly with the exteriors, for instance, adhesion forces, when you have these transmembrane molecules which are actually bound to the cytoskeleton and to the exterior, which can exert forces. So, and this has to be solved on gamma, on the interface. Then in order to solve this, of course, in general, you cannot access sigma from that, and anyway, you want to access the deformation too. So you need to define a rheology of this surface here, so of the actomyosin surface, and this will relate stress and strain, and we will have a look at that in what follows. And then, of course, that should be complemented with a force balance in the bulk, which I will simplify to perfect fluids for this talk, and some model of these adhesion forces, which is not obvious either, and I won't consider that either in this talk. Right, so for the rheology, that was the subject of my talk in September, actually, so you can actually have a look at the video online on the Newton Institute website. I have validated this in the case of single cells. So this is Maxwell-like rheology. If you set tau alpha and sigma a to 0, you have a viscous liquid with some compressibility. Okay. Now, if sigma a is non-zero, that's mimicking the effect of myosin. That's bringing something like a negative pressure, which is contracting the whole thing. So you have a viscous active fluid, which is contracting itself whenever it can, so that there is a pre-stress. You can understand that as a pre-stress in the sense of contraction. And here is the elastic term in the Maxwell rheology. So that's bringing the short time elastic behavior of this liquid-like actomyosin. Okay, so if you do an experiment at very short times, you'll be left with something which is elastic and has some compressibility and some shear resistance. Okay, so now we can try to write the full model now that we have the rheology. So the simplifications I'll use is that I assume that the bulk behaves as a perfect fluid, so it just preserves volume. That's a first simplification, so that there is a pressure jump between interior and exterior. And the adhesion forces in the setups I'll consider appear only through boundary conditions when they appear at all. Okay, so the full problem is to find actually the surface of our cell or embryo, find the shape evolution in time, the velocity, which is related, and the stresses, and also the pressure jump between the interior and the exterior as a function of time, all that, such that, well, the stresses obey the rheology I've prescribed, with here this source term which is providing energy. So that's a big difference between engineering systems and this living system, that the energy is input in the bulk, everywhere on the surface gamma. Whereas in engineering, usually you use the boundaries to provide energy to the system and to act on it. Then you have the force balance here, where I have simplified, so because I have a perfect fluid, I only have the pressure jump to balance in the normal direction, but I have to worry about the force balance in the plane, of course. That's the most tricky part. And then this is the volume constraint, so there is no net variation of volume. The normal velocity average to 0, which is, and the pressure here is the Lagrange multiplier corresponding to that. And my surface evolves with this velocity. Possibly in, well, only the normal direction, of course, will be important there. Okay, and then, okay, I've used these notations that the rate of strain tensor is the symmetric part of the velocity. There is a projection tensor here, so that's something like the identity on the surface. And this is the upper convected Maxwell derivative. Okay, so now I'm ready to try to solve this system on a few specific cases. So as I've told you, the first limit I'll consider is the short times elastic limit. So this means that if you non-dimensionalize that with respect to time, this term will become negligible. And so it will be the time derivative here, the objective time derivative of the stress tensor, which takes all these terms. And actually, if you integrate this in time, you find the classical linear elasticity equation. If in addition you assume that this tensor here of the myosin activity is isotropic, so this is the myosin is active in the same way in all directions of the tangent plane. Then you can combine. Well, then you can explicitly integrate and obtain sigma, and input that into the mechanical balance equation, and you find something which is really an equation for linear elasticity with here a pre-strain. Well, you can consider that as a pre-strain, so a preferred area, which is not the area of the initial configuration, if you will. The reference configuration is not the initial configuration because the myosin has imposed some pre-strain on the cell apices, okay, or on the cell surface. And you have the bending force here and the pressure jump. And this can be rewritten as the minimization of some elastic energy here. I don't and a shape optimization problem for that. So I'll tell you now of one application of this. That's the case of the gastrulation, so the formation of the ventral furrow in Drosophila. That was the first movement I pointed on the movie of the whole embryo. And here we have two views of the same, except that the ventral side is up, so this one is lying on its back. And you see from the top, and you see here a cross-section of the cells. So now focus on the green part. You see that it's forming a fold, and then it's rapidly invaginating. So you really have a flow inwards of the tissue. Well, I won't say a flow because I've said I was in the elastic limit, so the times are short-ish. These are minutes, and you see that the invagination, the real fast part, is lasting for less than a minute. So we can argue that we could use the elastic limit.
It looks like this tangential motion.
There is tangential motion initially, yes, sure, yes.
Are you taking this into account in your model?
Well, there will be tangential motion, tangential deformation, so that will be the elastic limit, okay? But yes, that's not just the normal motion. It's really elasticity. The shear elasticity is taken into account in the modeling. So there have been many models in the past, in the past 35 years, which were trying to reproduce this. Most of them are 2D and some of them are 3D. Although they all rely on not a thin surface, but a thick surface. So they were modeling, they were considering that the mechanical structure was the whole of the thickness of the cells here. And it's true, it's not a thin shell if you consider the whole thickness of these cells. But then the question is whether this is the mechanical structure that is relevant, or whether it is rather only the apical surface, so this surface here, where most of the actin and myosin are found, actually. And actually these guys here had put tracers, Lagrange tracers in the flow, these red beads, and they compared the streamlines obtained from these tracers with what you would expect if you impose these surface movements that you were noting, annotated, you impose them as boundary conditions of a Stokes problem. If you solve the Stokes problem with that, you obtain streamlines which are very close to the experimentally observed motion of these beads, which means that the interior of the Drosophila, at least in this ventral region where they performed the study, is behaving like a passive viscous liquid. So if it is a passive viscous liquid, it is not an elastic structure which is giving the shape of the cells. So what is giving the shape and what is important is actually the surface of which they just imposed the motion in this paper here. Okay, so based on that, we tried to reproduce the gas relation with simply an infinitely thin shell of elastic material, because we consider that we are in the elastic limit of short times. So we are minimizing this energy while trying to find displacements that minimize this elastic energy with a pre-strain for the cells which are in this ventral region here, marked in red. Okay, this is done with surface evolver, and the people who've done this with me are in Grenoble. Actually, so you start, so this is a section, this is actually a cut, seeing from inside, from a cut in the middle of the embryo, and as you make this evolve, so gradually you reduce the preferential area of the cells in this ventral region here, and this invaginates and gives some shape, which has some invagination. It's not the best reproduction of the actual Drosophila embryo across all the possible models. We believe that we have to impose more strictly the volume conservation, and also the existence of here that is a cuticle, so a membrane which is external to the embryo and which constrains its shape. So we believe that's the key to obtain something looking a bit more like this. But still, so really the interest there is that having an infinitely thin shell can allow you still to obtain the gastrulation of the Drosophila. And the mechanism for this is actually that you tense, it's a bit like tensing some cable on the surface in this direction. So this is really a 3D motion that we have here. Whereas most of these models, even those which were run in 3D, would actually work on a 2D system, simply on a circle. So that's a specificity. Okay, so I'll now turn to the second case I wanted to consider. So that's the full problem, but on a known shape. So if the shape gamma of t is known, then I reduce my problem to only two equations. The rheological equation and the equilibrium equation. And I'll do this for this problem. So that's an experiment I have presented already to you in September. So just a quick reminder. So my colleagues in Paris, FFS Nassios and colleagues, were grabbing cells which were still spherical when suspended in a liquid. They were grabbing them between two plates, which were coated with fibronectin, so the cell would adhere on both plates. And then they would maintain just the end of the two plates, so they only provide some stiffness, and the cell can bend the more flexible plate, and this is what it does. So it spreads on the plates and it deflects the plates. And we can record the height of this, the deflection of the plate, and knowing the stiffness, we also record the force that the cell has been exerting on it. Okay? So in this problem, the boundary conditions are given in terms of the force that we measure in the z direction here. So if I integrate the force along this direction here and take the scalar product with the z direction, I obtain the force in the z direction here. So that's sketched here. The shape is simple, so I can write this simple model of the shape where I have arcs of circles at a distance RC. So this is not a generalized minimal surface, by the way. The radii of curvature don't agree with that, so it's more general than that. But still, it can be parameterized with just 3 parameters here to define this full shape. Okay, so now, thanks to the symmetry of this object, one can actually solve this equation before worrying about that one. So because there is no shear stress at all because of the symmetry of the problem, I can actually, so this is how the equilibrium writes in the curvilinear coordinates along this surface here. And one can actually solve this and obtain sigma, the stress in the longitudinal direction along the curvilinear coordinate here, and sigma orthogonal, which is the orthoradial stress from this balance. And you get that as a function of the force in the z direction and the pressure. And then, knowing this dependence of sigma, so of course it also depends on z. It doesn't depend on the theta coordinate. No, it was phi. Yeah, it doesn't depend on the phi coordinate here, but it depends on z. And then knowing that, you can turn to the rheology equation. So here you have 2 equations for the 2 in-plane directions. And you can actually look for a solution of them, which is nearly accessible analytically, although the mechanical problem is a bit different from what I am mostly presenting here, because the unknowns are not the ones that you would think they are. So maybe I won't go into too many details, but to look for it was possible to look for the equilibrium shape of that. So guessing what were the height h and the pressure and the z force when you have equilibrium, so when the shape is not evolving anymore, but there is still a surface flow, which is a backward retrograde flow on the surface, which is balanced by a polymerization at the end. But still, so that's an example in which having a simple shape allows you to solve these equations in their full complexity on that surface. But that's not something that you can generally do. So that's why the next part. Okay, so this is the comparison with the experimental data across many orders of magnitude of external stiffness, so the stiffness of the plates we're using. So we could do something quite quantitative there, and with actually, so there are 4 parameters to fit for this and the dynamics. So there's quite a bit of prediction here, actually, in addition to fitting. So, okay, so that was a simple shape, but what about complex shapes like this one? And can we do something like this? So for the time being, I don't think we really can solve viscoelastic equations on that surface. But I'll show you the viscous limits on that. So maybe just again the early morphogenetic movements in Drosophila. So this is seen from the side. The ventral side is here. And I will focus now on the germ band extension, which is the flow of cells. So that's here now. So initially you see cells going down towards the ventral side, and that's the ventral furrow formation, so they are actually going in there, and then they extend, they go in that direction, so you have really kind of vortex going in that way. That's called germ band extension, and it's essentially a tangential flow. So the shape doesn't change, it doesn't invaginate at this, in this place at that time of the morphogenesis. So there has been a lot of work, of biophysical work and biological work on the causes of this germ band extension. So we know that the germ band, actually this part of tissue which is extending, which is flowing in that way, expresses more myosin than the rest of the tissue. And we know also that this myosin is not acting in an isotropic way this time on the surface. So here you can probably see at the end of the arrowheads that the myosin is recruited much more on the vertical edges, so the edges of the cell which are in the so-called dorsoventral direction, and there is much less on the anterior-posterior edges of the cells. So this has been tested by a number of groups. It's now well established, although for us maybe it's a bit dubious to rely on such images, but this has been quantified, and we know for sure it's so. So this means that the source term for myosin activity should be also anisotropic. So rather than being something times some quantity times the projection tensor, it will be this quantity times the cross product of the dv direction vector here. And then it's located mostly on the ventral side, and we will use this anisotropy and try to reproduce this flow that we observe. Okay, so this is a flow which is tangential to the surface, so I assume that gamma, the surface, is known, and I want to find, now the velocity is denoted u, sorry, it's not v anymore. I want to find the velocity, the stresses, and a Lagrange multiplier that forces the flow to be tangential. I observe it's tangential, so I solve it in these conditions, but still I need to impose it mathematically to keep u tangential. And so this is my new system here, and sigma a will be this anisotropic guy I've shown you. And this is actually joint work with people in Grenoble in the Math Institute. So we neglect the elastic terms for now because we're not able to treat them, and also because the flow is lasting for 90 minutes, so it's a bit long compared to the relaxation time that I found in previous projects for actomyosin, although that could be discussed. So possibly this term has some importance, but we cannot deal with it for now. And we write this as a constrained minimization problem, which is possible also in that case because we have an explicit way to write sigma, so we can put it into the force balance equation. So we want to minimize the viscous in-plane viscous dissipation plus here it's the the surface area variation dissipation, and we have some source term here which is based on the divergence of the myosin term, and we need to solve this in the space where the velocities are tangential, and we need to impose that they are tangential in H1, else we will have, if we impose it only in L2, this will not work. We will have some zero eigenvalue in the problem. So that was something that took us some time to realize, actually. Okay, so this is the constraint minimization problem, and in order to solve it, we use a Lagrange multiplier approach, where we have this Lagrange multiplier, which is now called theta. That was F tangential a little earlier, sorry. And we solve this, we want to solve this saddle point problem. So this is the energy to minimize, and we add this Lagrange, well, we have this Lagrangian here with theta appearing, and we can have some coefficient actually to fix how tangential our solution should be, but we can take gamma to infinity without a problem, so that's the limit case here. Okay.
You haven't given us what is sigma a of x, the function, the source term function.
Oh yeah, it's, well, I have just sketched it. So this is something. There is an indicator of this domain times these this cross product of vectors, which are also defined in a complicated way. So they have to mimic something like that on this ellipsoid. Okay, so that's, well, that's a bit of a cooking recipe for that. But that's just a source term. So it has to be in heterogeneous in space and anisotropic in order for things to work. Well, in order for the flow to be interesting. That's, I can solve, but that's not an interesting flow I obtain. Okay, so don't think we're missing anything now. So this is the way you solve the saddle point problem. So you have, well, you have this this term linking the velocity. That's the in-plane dissipation. There should be, yeah, and we also have used a Lagrange multiplier for the compressibility, so we can go to the incompressible limit also for these flows. So that's here the compressibility terms, that's the divergence of the surface velocity, surface divergence of the velocity, and the Lagrange multiplier associated with it. And we can have here this lambda, this compressibility modulus. Okay, now that we have these equations, we need to solve them on an approximation of the exact surface of the exact gamma. So this is quite classical, and people around Anotida and people in Warwick also have been doing things like that with scalars for a long time, solving these geometric PDEs. So here the difference is that we have to deal with vectors and tensors on that surface. One step is to approximate gamma with gamma h, which is an isoparametric finite element mesh. So you start with a triangulation like this, and you construct some higher order approximation of gamma. And on that we take the velocity, the surface pressure, and this Lagrange multiplier for tangentiality in these finite element spaces. So we have, for instance, for k equals 2, we have P3 velocity, a P2 pressure field, and pk, so a P2 tangentiality Lagrange multiplier here. And this turns out, so we have the numerical analysis of this in the, not in the saddle point problem, but for the penalized case. In the for the full case of the Lagrange multiplier approach, we have only empirical convergence, which is sketched here for k equals 3, which is more or less the best compromise we had. So that's quite high order finite elements that you need to use, because you want to approximate well the curvature of the original surface. But this converges. So we can go to applications now. So this is the mesh that we've designed for the Drosophila, with here, this bit here, you maybe haven't seen that well in the previous slides. It's called the cephalic furrow. That's a region which is already invaginated at the start of the German extension. So that's actually the most difficult part to mesh. The rest is a deformed ellipsoid, actually. Then we impose the patterning. So here, really in this talk, I assume that I know how the patterning is being generated, and I put Myers in this location. And I solve for the flow, and this is the flow I obtain, where you have really these vortices on the surface. I have a stagnation point which is not in the actual embryos. I have to work on that a bit. But definitely you have some what is called convergence. So this is these cells here are flowing towards the ventral side, and extension, so you have a flow towards the posterior end here. So we can reproduce somewhat what is observed in nature, but we're very far from quantitative. So at this stage, we could also wonder, okay, why are we doing all this? This is a complicated numerical method that I've introduced. I've had to make a number of assumptions and simplifications. I'm not even getting something which looks too close to biology. Well, the first use, I think, is that we can rule out some modeling assumptions. So when we get something which cannot agree with the biology, then we can think, well, this modeling assumption was wrong. So I think that's the first thing, that we are not validating our assumptions, but at least showing that they are still consistent and they are not in contradiction with the biology. And we can also test the effect of some variations. So we can, even if we don't obtain the correct flow, we can say, what if? And for instance, we can say, what if we didn't bother about this annoying cephalic furrow, which is so difficult to mesh? And I guess I don't need really to show you the flow pattern for you to guess what happens. This is all symmetrical now. So of course, the flow will be symmetrical too. And here it's not. It's known in biology that it's not symmetrical, whereas the patterning is more or less symmetrical. So maybe I could have said it without running the simulation, but now what I can say is that this geometry has probably an impact on the flow that is observed in biology. And this is something which is not so well established, and that hasn't been said in so many words, I think, so far. So that's actually the dogma of biology. You go from genes, then you have proteins, and the proteins set the morphology. Well, it's been recognized that it's a bit more complicated than that. So first, you have all these feedbacks occurring, and also from going from proteins to morphology, people have realized that mechanics was really important, and that you needed to worry about that. That was not simply, the proteins give the shape. And also the role of the environment on the gene expression is something which is very well established. What I'm saying is that the morphology that you've obtained in the previous morphogenetic movement can have a feedback on the mechanics directly, not going through the expression of different genes, but simply the fact that you have some mechanical boundary, some barrier to the flow, can generate a different morphology in the end, because you flow in another way. And actually, the other part that I had shown you about these cells, which are reacting to the boundaries, is also something that can complement this picture. So there we were varying the stiffness of these plates, and through mechanical balance only, we were obtaining different morphologies for them. So maybe if I have one more second, I can show you how this was done. So we were varying the external stiffness, and we were going from this phenotype of very flat cell to this phenotype, as biologists say, of very tall cells. And this is complementary to what happens from a biochemistry point of view, where if you change the amount of myosin using drugs, you will get different phenotypes also. So you have two ways of balancing the of getting some phenotypes. You can vary the environment, the mechanical environment, or you can vary the gene expression, and that's actually what I had here. So you can do it in two ways: changing the proteins or changing the environment. Okay, so these were my biology conclusions. Now a few mathematical conclusions. I think the biological model that I've shown you, well, does have some relevance for both single motile cells and embryos, which is quite good. So early embryos lack extracellular matrix, which is much more elastic, so which is helpful for that. So we can do some numerical or analytical resolution in some cases and learn something from that. But I think definitely there was a big need for this whole program, because we need new mathematics. We need to be able to solve more on this field, among others. And with that, I thank you.
Thank you very much to the speakers. And are there any questions?
Could you comment about the bulk dynamics?
In general, or in single cells?
No problem. Don't you think that the bulk dynamics will have a say on what's happening on the surface? So you've taken surface PDs, which is great. I think it's very nice.
Mm-hmm.
But do you have any comment on? Is there any experimental evidence to suggest that the bulk dynamic properties could be interesting?
So, well, yes, it's. Okay, so there is this experiment where they show that it behaves like a viscous liquid, which doesn't say that the dissipation in this viscous liquid is less or much less than in the surface. That has still be this still has to be shown, and also they do it only at one time during the development and in one location in the embryo. So there remains some need to really show that this is the case all through the early Drosophila development, and I'm not sure this is the case. I think first you have more myosin in the basal site, so that's the inside of the cells in the cut. Yep, it's called basal. Yep, I have to go back. Sorry. Yeah. So yes. So here, close to the place where the ventral furrow invaginates, you have next to no myosin here, but on the other places around there is much more myosin, which is left from a previous movement, but it's not sure whether it has a role or not in the flow. So it may be that there are several shells, for instance, one in the other, or something like that. So it's something that still remains to be explored, and I think numerical.
Also, could you comment on the acting? So you have taken a function, but I would assume that there's a dynamical process of how actin is distributed within
myosin, you mean?
Yeah, myosin.
Yeah.
Yeah.
Okay. Yeah.
Would you consider coupling viscoelastic to some dynamical process model for myosin?
Sure. Yeah. That would be. So that's something that we have that we've started to do. So at a smaller scale, at the scale of the cell, really. Then, because you have also a dynamics of the myosin which is happening at a subcellular level, and there we can hope to do something about that. Yeah.
I think it's a time we've already got 5 minutes passing to Stephen's time. So
okay.
Let's thank the speaker again.
Thank you.